In this video, we will discuss about mechanism of action of enzymes. Enzymes are biological catalysts that speed up chemical reactions in living organisms without being consumed in the process. The mechanism of action of enzymes involves lowering the activation energy, enzyme-substrate interactions, thermodynamics of enzymatic reactions. The first mechanism is enzymes lowering the activation energy. Then what is activation energy? It is the energy required by the reactants to undergo the reaction. It's the energy barrier that reactants must overcome to reach the transition state. Transition state is high energy state where bonds are breaking and forming. Without the help of enzymes, reactions often need high temperatures or other energy sources to push molecules over this barrier, which are impractical in biological systems. Next is enzyme-substrate interactions. There are a few theories that explain the mechanism of enzyme action. The first one is lock and key model or Fisher template theory. It states that enzymes have rigid structure with binding site that only specific substrate can fit into, like key and lock. The lock describes the enzyme, specifically its active site has fixed, unique shape determined by its protein structure, whereas the key describes the substrate is the molecule with complementary shape that fits perfectly into the active site. Only the correct substrate can bind to the enzyme, much like how only the right key opens specific lock. However, this model does not explain enzyme flexibility and transition state stabilization. The next one is Koshland-induced fit theory. It states that enzyme-substrate complex formation involves induced fit where the substrate interacts with the enzyme, causing conformational change that strengthens the binding site and enables catalysis. The binding of the substrate to the enzyme's active site induces conformational changes in the enzyme shape, similar to glove conforming to hand. The Michels-Menten theory describes how the rate of an enzyme-catalyzed reaction depends on substrate concentration. The key idea is that an enzyme binds to a substrate to form an enzyme-substrate complex, which then converts to product and releases the enzyme. The next one is acid-base catalysis. The enzymes enhance reaction rates by using amino acid side chains to donate or accept protons, stabilizing transition states, or facilitating bond breaking or making. In substrate strain theory, the enzyme distorts the substrate into high-energy conformation resembling the transition state, reducing the energy barrier for the reaction, such as in lysozymes hydrolysis of glycosidic bonds. In catalysis, the enzyme forms temporary covalent bond with the substrate, creating reactive intermediate that accelerates the reaction, exemplified by the serine protease mechanism involving covalent alkyl enzyme intermediate. In the entropy effect, the enzymes enhance reaction rates by decreasing entropy and straining substrates to the transition state, increasing collision probability and reaction velocity. The last mechanism is thermodynamics of enzymatic reactions. The enzyme-catalyzed reactions may be broadly grouped into three types. In exothermic reactions, the energy is released or liberated from these reactions. In isothermal reactions, the energy exchange is negligible and the reaction is easily reversible. In endothermic reactions, the energy is consumed in these reactions. If you found this video helpful, please like, share, and subscribe.